thank you for your attendance, and no matter where you are in the world and when you happen to be watching this. It's my pleasure to be invited to provide this virtual distinguished lecture on a topic that's of a great deal of interest to me. And I hope that you'll find it interesting as well. So let me just share my screen with you. I hope you can now see a slide showing a heart beating. And unless I hear Judy tell me otherwise, I'm going to assume that that's what everyone can see. So I'm broadcasting here today from the University of Auckland in New Zealand. New Zealand is, of course, tucked down on the bottom of the South Pacific. And the University of Auckland is New Zealand's largest university. And we have about 42,000 full-time students, and we live in a city of about 1.65 million people. Now, what I'm going to be talking about today is a summary of work that we have done to try to understand better the heart. And in particular, we're applying the principles of instrumentation and measurement to understand how the heart functions as a machine. And here I'm referring to it as nature's engine. And so what you're looking at on the left there is a heart that's been kindly donated by a rat. So to give you a sense of scale, that's about the size of a human thumb in size. So perhaps 10 to 15 millimeters wide by maybe 20 millimeters tall. And to the right of that is a single muscle fiber, referred to as a trabecula, that has been extracted from that heart. Now, of course, we all rely on our hearts for life, and the heart is a natural engine, and it is comprised of many billions of tiny muscle cells that each are their own intact engine. And so what I'm going to talk about today is applying sort of principles, I suppose, of mechanical engineering to the study of the heart and of its tissues. Now, when you have an engine, such as an internal combustion engine, and you want to study it, you would connect your engine up to a device that is commonly referred to as a dynamometer, hence the title of this, this talk today. So a dynamometer is a device that can be attached to an engine and can apply very controllable loads to that engine and measure many of the parameters of interest that define the performance of the engine. So in the case of an internal combustion engine here shown on the left, we might wish to connect to it a device that allows us to apply loads to the engine to put it under duress and then measure its performance as it provides different torques, for example. And we might measure a whole bunch of different parameters related to the power output of the engine, the speed at which it is operating. And so these same principles can be applied to the heart, which of course is a completely different type of engine. It's entirely organic. So let's think for a bit about the things that, if we were to apply the principles of dynamometry to a heart, what are the things that we would want to measure or to control? Well, first of all, any engine needs fuel. And so for the heart, that fuel comes in the form of a chemical called adenosine triphosphate. And that is cycled continuously in the heart's tissues with the use of oxygen. And so if we could measure adenosine triphosphate, commonly referred to as ATP, it's abbreviated, or the amount of oxygen that the heart is consuming, that tells us how much fuel it is consuming as it beats. And of course, that consumption of fuel, or the performance of work rather, is initiated electrically in the heart by an electrical impulse, a bit like in your internal combustion engine, the spark that ignites the fuel. So we might wish to control both of those two things, measure the fuel consumption and control the electrical stimulation. In terms of loads that the heart experiences, well, the heart has to drive against a mechanical impedance as it tries to pump fluid around the body. It has to develop pressure and that pressure has to overcome the impedance that is presented to the heart by the circulation of the body, whether that's the pulmonary circulation or the peripheral circulation. In doing so, that load has work done upon it. So the blood, as it is pushed around the body, is the subject of work done from the heart. That leads to an increase in temperature, a very slight increase in temperature in the blood. So temperature is something we may wish to measure and control. And we may also want to load the heart with drugs to investigate how its performance changes as we apply these loads upon it. In terms of outputs, well, the heart, of course, produces pressure primarily, and that pressure induces flow as the pressure is applied to the blood and the blood exits from the heart around the circulations. There's also the heart rate, which is analogous to the RPM of the engine, if you like. And then there's the amount of work that the heart does as it beats and the amount of heat that is given off as it does so. And finally, there are some interesting properties that we might wish to measure of this wonderful organic machine. Perhaps we're interested in the stiffness of the heart's tissues, because these can change during diseases of the heart and can affect its performance quite profoundly. We might also be interested in its geometry, which again affects its performance, and its efficiency. And all of these three things, these three properties, can be affected as a heart is deteriorating because of some sort of disease or other. So before we talk in any more detail about the heart and how it works, let's overview what a combustion cycle, if you like, in the heart looks like. Now, of course, in the heart, it's a regular single, essentially a single cylinder engine, although it's got two ventricles. You can think of them independently. They fire in succession. And this is the cycle that goes on within each of these ventricles, each of these cylinders. The heart initially has a certain volume, which is referred to as its end diastolic volume. When an electrical impulse is created in the heart's tissues, that impulse induces an increase in pressure. So the pressure increases, and then once that pressure is sufficiently high to overcome, first of all, to open the valve and overcome the pressure of the circulation, the heart will now begin to shrink as it ejects blood out through the circulation. And so that reaches a point where it can no longer supply that pressure, and then the valves close, and then the pressure diminishes, and then the heart, that's referred to as the end systolic volume, then the heart is restretched by incoming blood, ready for the next beat. So this is one heartbeat, and there is an area enclosed by this pressure volume loop. This is referred to as a work loop, and it's very important in studying the heart, as we shall see. The heart can describe all sorts of different work loops, some with higher pressure and smaller ejected volume, and some with lower pressures and smaller and larger ejected volumes. And so these are a family of work loops that you might measure from someone or some animal under different conditions, perhaps different exercise conditions, or perhaps when there is some sort of disease or other. So the bottom red line here traces out a relation which is referred to as the end diastolic pressure volume relation. You can think of that as sort of the passive elastic properties of this big balloon. And then there is this other curve here, which is referred to as the end systolic pressure volume relation, which really describes the contractile and pumping performance of the heart. Now, it's important to note that the area of each of these loops represents the work, or is in fact the work done on the blood. This is a pressure volume plot. The area enclosed by a pressure volume plot is work done, analogous to a force length loop in the one-dimensional case. The efficiency of this engine, like any engine, is the work done on the load divided by the energy consumed. Now that's a very interesting parameter, an important parameter that we might wish to measure, the efficiency of the heart. And so I'll go overview today how we control the work done and measure the work done and the energy consumed. So here's an example from an intact heart. the rate of energy consumption, and this is, I promise you that this slide is the only one I think that has any mathematics on it, and they're just equations. So the rate of energy consumption, or which is here referred to as the change of enthalpy, is proportional to the amount of oxygen, the volume of oxygen that the heart consumes. So if you want to measure how much energy is being consumed by an intact heart, you can attach tubes to each of its major vessels and measure stuff as it goes in and out of the heart. Now, typically with a heart like this, we've replaced the blood that would normally circulate with an oxygenated saline solution, which provides all of the nutrients and oxygen to keep the heart alive for many, many hours. So if we cannulate the arteries in the heart, we can measure the volume of oxygen that it consumes by measuring the partial pressure of oxygen using oxygen probes up and downstream of the heart. And because we know something about the amount of oxygen that can be dissolved in the solution, we can measure how much oxygen is being consumed by the heart as it does work. So we cannulate these vessels, and that tells us the oxygen that is consumed. Now, because we know what the energy density of this oxygen is, it's 20.1 megajoules per cubic meter, we can now measure the change of enthalpy as being the product of the energy density and the volume of oxygen consumed. The efficiency then is the work that's done by the heart, and we'll talk about that in a moment, divided by the energy that it consumed, and that turns out to be something like 25%. That is, 75% of the energy that is consumed by the heart is not turned into work. Only 25% is done on work is done on the blood itself, which is an interesting thing. So we can rewrite this equation of enthalpy in this form. We can say that the change of enthalpy, which is the amount of essentially energy that is consumed in total by the heart, can be given by the rate at which work is being done, and the rest of that energy turns into heat. So this is now the heat rate, Q dot. And so that's an alternative way of measuring the efficiency of the heart. You can take the work done by the heart and divide it by the sum of the work and the heat, and that is also a measure of efficiency. And it turns out that this method is the one that we use if we wish to study individual muscle fibers that are taken from the heart. And this is our preferred way of studying the heart tissues because we get a lot more information about what's going on. Inside each heart, and indeed inside our hearts, there are these rod-like structures. They're cylindrical, they're about the diameter of a human hair, and they're about, say, 2 millimeters long. And they're one-dimensional contractile elements that can consist of many, many muscle cells, but they can be excised from the heart. You can even get these from humans. And this is what they look like when they lie in a dish. So they're usually, well, good ones look like this one here, which is about 100 microns in diameter and perhaps 1.5 millimeters long. So they're really useful specimens to study because the contraction is entirely one-dimensional or predominantly one-dimensional. So what we do is we take these little one-dimensional specimens and we mount them between two hooks so that we can now hold either end of the muscle and study the little engine that it is very, very carefully with a range of different imaging systems and measurement systems. So this is now, let's now talk about, think about what we would want to measure if we wished to measure the performance of this one-dimensional actuator from which the heart is constructed. So there are mechanical parameters we might wish to measure, such as length and force and stress. So that's here, I'm showing an example where we're stretching out the muscle, and you can see it increases in force. The force vector is shown here as this black arrow. Once we've stretched it, we might then electrically excite the muscle to produce force. So we give it a little pulse of a small electric pulse of a few volts, and that's enough to make the muscle twitch and produce force. On the other hand, we might want to produce an isotonic twitch, and that's when we adjust the length of the muscle so that the force that it is driving against or producing remains constant as we electrically excite it. We can measure stiffness by wiggling one end of the muscle and watching the resulting change of force at the other end. And we can also control work, which I'll describe shortly. Now, in terms of the metabolic parameters of the heart, we would like to know how much energy it is wasting. And so to do that, we measure the heat rate. And that's done by, we have to keep this muscle alive by bathing it continuously with oxygenated saline solution. And that solution warms up ever so slightly as the heart does its work. The temperature increase of these muscle specimens is only about 1 millikelvin. That is, as we wash water along this muscle, oxygenated solution along the muscle very slowly to keep it alive, that water will increase in temperature by 1 millikelvin. But that temperature increase is proportional to the rate at which the muscle is losing energy in the form of heat. We can also measure oxygen consumption, which is the other way of measuring energy consumption. We can also look at the sparks that are firing the engine. We use calcium imaging systems. Calcium is the ion that is predominant in cardiac tissue that engages the contractile engine so that it produces force. We can measure that using fluorescence techniques. We might be interested in measuring how much the muscle is shortening, and we can do that by examining the proteins inside the tissue using some imaging systems that I shall speak about. And from these images, we can compute the strain inside the engine and take into account its change in shape as it contracts. So here is an example of a three-dimensional image of a cardiac trabecula. It's just a stationary image here. This one would be slightly less than 100 microns in diameter and about 2 or 3 millimeters long. This is created using optical coherence tomography, and I'll show you some images of that shortly. And these are some platinum hooks that we use to secure either end of the muscle. So let me present the instrument that we have built, which we refer to as a cardiomyometer, for studying these tissues. So first of all, let me orient you to show you that the muscle fiber is measured, is mounted in this little small glass chamber right in the center of this gold block. And you can see some laser light that's firing through it at the moment. So to give you a sense of scale, this is about 40 millimeters from here to here. So in order to create this dynamometer, these are the things that we have. We have first of all, we have an imaging system. So this is a condenser that shines light down through the muscle in the vertical direction. And there is a corresponding microscope objective hidden underneath that allows us to do calcium imaging and to look through the muscle as it contracts. But in order to control the length, we use a laser photometer. First of all, we split the laser beam, and this laser is focused down onto a cantilever. That cantilever allows us to measure force production because the cantilever is actually attached to the end of the muscle using a small glass tube with a platinum hook. Another interferometer beam comes through this path here, and that measures the end or the position of one end of the muscle. It's attached to a linear motor. And then a third beam is bounced off another voice coil linear motor at this end. Then we have an OCT scanner, which we've built to examine the three-dimensional shape of the muscle as it contracts. So this is what we refer to as our cardiomyometer. We take these small samples from the wall of the heart and we insert it into the instrument, into this glass chamber. And here's not a very pretty sample. This is a terrible one, which I always mean to update. But again, you can see that muscle, you may have seen that just contracting as we electrically excite it. And that was running in what we refer to as isotonic mode. So as it contracts, the muscle is shortened by a control system to keep the force constant. So our device uses these two linear motors that hold either end of the muscle. And these are all controlled by a laser interferometer, as I've shown, based feedback control system running in LabVIEW FPGA and LabVIEW real time. And so that enables us to now stretch our muscle and translate it backwards and forwards in the view of our imaging systems. If we look down on top of the instrument with a close-up view, here's our two hooks. We're looking down on top. And you can see here, we're just changing modes to show you that the motors that control either end of the muscle can now stretch, they can translate, they can contract. And in the center, these red lights and purple lights indicate the imaging system. The purple is the calcium imaging system, and we do just white light microscopy also through the same system. And the OCT system is orthogonal in the direction of the red laser. The OCT, the optical coherence tomograph, gives us the three-dimensional shape. So I'll talk about these systems in a moment, but this is the sort of data that we get from it. So here's the calcium transient. So this is the spark of ions that flood into the cell when we electrically stimulate. And so there's a number of beats here. It's like firing your engine at a certain amount of, at a certain hertz, a certain frequency. And here's the force that the muscle fiber produces, and that's in the millinewtons range. So maybe maximum of 10 millinewtons for a typical specimen. Notice that the force lags the calcium ions flood into the cell, and then the force builds up. And as the calcium ions die away, the force also dies back. At the same time, we can image the strain within the tissue. We can measure its shape in 3 dimensions. And in fact, in 4 dimensions, as the muscle is twitching, we can get the full 3-dimensional shape throughout a twitch. So how do we make all of these systems work, and how do they work together? Well, I've talked briefly about how we measure force and length. Let me talk first now about how we measure the shape of the muscle. So here is a slightly different bath, but here's the glass bath in which the muscle sits, and we're looking here at a infrared laser. It's being shown here on a CCD camera that can, or CMOS camera, sorry, that can resolve down into the infrared. And this is an OCT, an optical coherence tomography system, that is scanning across the cross section of the muscle. And so here is the muscle cross section. It's slightly oval in this case. So we can measure, you can see here the laser scanning that piece of tissue, and this is the tissue cross section. And we can watch it as it contracts repeatedly, twitching over and over. But because we can now translate our OCT system, you'll see here the laser is scanning along the length of the muscle. We can build up over many identical twitches a series of cross sections of the muscle contraction and build these into a full 4-dimensional representation of the muscle contraction. So here is an example of one muscle that has been stretched out. This is its initial shape at the bottom, and here we've taken that same sample and we've stretched it out to its so-called optimal length. Optimal length is the length at which the muscle cells produce the maximum force without too much increase or significant increase in its passive elastic stiffness. So that's done with this 4D gated imaging technique, where we gather one cross section per twitch, but we scan along the length of the muscle, perhaps over a period of 10 seconds, to get this 4-dimensional data set. And that gives us rich information about the shape of the muscle during a twitch. That one's rather boring. Some of them are a bit more interesting. This one here, for example, you can see has a little more interesting shape. It's got this wisp of tissue hanging off it, and another one on this side, as we're here rotating the OCT view so you can see the full 3-dimensional shape. This muscle's very short and fat, and you can also see artifacts coming from the reflection of the OCT system off the hooks that are holding the tissue. But because we've got this information, we can now do interesting things like track the motion of different parts of the muscle over beats. And here you can even make out a wave of contraction going from the left to right. If you just imagine it or squint, you might see a sort of a wave of motion coming out here. This is really unique and interesting information that until we did this, no one had done on cardiac muscle in this way. So that's how we measure the shape of the muscle, which is an important part of how it's performing. But I did mention that we were also able to image down vertically in an orthogonal direction through the tissue. Now that's using, in the direction of this arrow, using a microscope, and the condenser for that microscope is above the instrument, where the collection objective is down below. And so if you look into cardiac tissue, you'll see striations, so stripes, which are created by the proteins, the contractile elements within the tissue. The dark bands and the white bands represent a series of little engines that are all tacked together in series. So we can see these in a cardiac muscle as it contracts. And so this is a muscle video recorded. It's slowed down here so we can see it through our imaging system. And you can see the stripy black lines and white lines if you look carefully. Now, of course, as the muscle is excited and contracts, those lines get closer together as the muscle shortens, depending on what sort of load you put on it. So that's one video recording. We can now move the muscle in our imaging system and get many different recordings of its internal contraction over time and stitch them together. So this is, again, taking a sort of gated imaging approach to build up a large, high-resolution image of the full muscle contraction. You can see some interesting things going on in this particular sample. First, there's a big blob of tissue at the end here, which we'll kind of ignore for the time being. But as you can see, when it contracts, when it is excited, some parts of the muscle are moving in contraction, while other parts seem to be stretching, and there seems to be this wave of activity going down this sample. So having stitched all of these images together, we can now apply some interesting image registration techniques. This is work that was done primarily by one of my PhD students in here, who has used or developed a technique for doing image registration to a very high resolution. So something down to something like 1 millipixel resolution of displacement measurement between adjacent images from a video recording or between two different images of the same object. So this is, we refer to it as subpixel phase-based image registration. Now, this technique allows us essentially to seed our muscle sample with a bunch of little spots that we can now track. These are just material points that are created in our software. We're tracking the centroids of these spots during muscle contraction. So this top plot is going to show the X displacement from the image, and the bottom is going to show the Y. So red means that the pixels are moving to the right, and blue means to the left. So as you can see, initially with this sample, both ends are moving towards the center. The left is red, it's moving to the right. The right is blue, moving to the left. And so we are resolving the motion of those material points to a very, very high degree of resolution. We can also track them in the vertical direction in the bottom plot. That then allows us to compute the strain, of course, which is it's not the normalized distance between samples. And so now we can look at the strain field as the muscle is excited, which provides very useful mechanical engineering information if we now wish to describe this muscle's performance in terms of its, and define its material properties and its contractile behavior. So there's a rich amount of information that we are gathering with this technique. Now, in case you're wondering, of course, this technique can be applied to images of any type and indeed any dimension, as long as you have features within it that our algorithm can track. And speckly images are perfectly fine, of course. These are stripy images, and our algorithm works very well at tracking those over time. It allows us to do very interesting things, such as look at the trajectory of individual material points. So the circle here is showing the resting position of a particular point in our muscle sample, and we are now tracing its motion as it contracts. And you can see in the case of this point, initially it fired off to the right until the peak of contraction, at which point it started to move back to the left, but also downwards. And so this is a really interesting technique for tracing out the motion of individual points within a sample and using that information to, or interpreting that information with the use of modeling techniques. So this is a different sample. In this one, we're also showing sarcomere length. Now that's the distance between the contractile units in the muscle. Each unit is referred to as a sarcomere. And so with this analysis, we can also, from these images, we can also measure sarcomere length as well as strain. So let me just show you that video again. And you can see that the two parameters, as you would expect, are highly correlated. The sarcomere length, unsurprisingly, really is very similar to the strain that's developed in the tissue. Okay, so we've now measured the shape of our sample, of our engine. We've measured its shortening behavior or its lengthening behavior. What else might we be interested in? Well, we're very interested in measuring the amount of energy it consumes. And for this, we measure the amount of heat, or sorry, the amount of temperature change of the muscle. The energy consumed is the heat. The heat arises from the inefficiency of the tissue, and it causes a temperature change in solution. So we have two thermopile sensors that we have put, one downstream of the upstream of the muscle and the other downstream. So we wash the muscle with solution. As that solution passes along the length of the muscle, it is increased in temperature, and our two thermopile sensors, which are actually just very small thermoelectric heat pumps used in reverse, they measure the temperature increase of that solution to around about 1 millikelvin temperature change and about 1 to 2 microkelvin of resolution. So it's an extraordinarily sensitive measurement to make. This is a finite element model showing a muscle producing heat. It's a series of step responses, and you can see that some of that heat is causing temperature change of this downstream thermopile sensor. So that's how we make this measurement. That allows us to resolve the heat rate to a resolution of 10 nanowatts. So it's a calorimeter for very, very small calories, or microjoules, I should say. Okay, so now we can measure the stress that's produced by the muscle and the rate at which it loses energy in the form of heat. And so you can see here that these two things are quite highly correlated. This black band is in fact a series of individual twitches. They're all jammed together, but you can see here that this is the active twitch force that's being produced by the engine on each twitch. And you can see how, just by visually inspecting, how related the amount of heat production is to the amount of force that is produced by the tissue. All of this sort of work has to be done inside a very thermally insulated environment, which we control to a high degree. No pun intended. We control to millikelvin, sub-millikelvin resolution to keep the experiment very stable. Right, so now let's get finally to sort of the dynamometer part in particular. So we've measured shape, strain, we've measured force, we're measuring the calcium spark that's turning on the engine or firing the engine, and we're measuring how much heat the engine is wasting. Now, what about the work that it is doing? So these are the stress time plots that you might see from a piece of muscle tissue or from a heart under different conditions. And these are corresponding length changes as that engine switches on or is fired. And typically, as I showed before, these are plotted as a work loop. So this is the stress on the vertical axis plotted against the relative change in length. And the letters here from A through to H are here shown parametrically as work loops. And so this is an experimental intervention that's commonly done on isolated hearts, and those who can will do it on isolated tissues to examine the performance of the engine. The area of each loop is the work done by the engine on each beat, and the heat rate measured by the thermopile sensors is a measure of the energy wasted. So now we can measure the heat rate, we can take the ratio of the work to the work plus the heat, and now we've got a measure of efficiency. And let me just skip to the important bit. this is what the efficiency curve of the heart and its muscle fibers looks like. This is the relative load that the heart is experiencing, and the peak efficiency of muscle fibers is typically around 15 to 16%. The whole heart is more like 25% efficient, because the heart experiences a load for which it is seemingly much more optimized than the loads that we typically present in instruments. But more on that now. So let's think about, if you have an internal combustion engine and you put it on a dynamometer, you might want to apply a torque load to measure how much torque the motor is producing. And of course, internal combustion engines will change the amount of torque and power that they can produce at different RPM, firing rates, and under different load conditions. Well, the heart is no different. So what we have done in our device is create a mathematical model of the impedance, the flow impedance that the heart normally experiences when it's in the body. And this is often, the circulation is often modeled as a so-called wind kessel, which is kind of like it's like a knapsack sprayer that you might use to spray weeds in your garden. You pump it up and it holds pressure, and then some of that pressure is bled out through a resistive element. So your knapsack, like the heart, has a knapsack sprayer, has a backpack sprayer has a valve on it to keep the pressure. So this is a model that's often applied to the circulation. There is a characteristic impedance of the aorta, there's the capacitance of your circulation, and there's the peripheral resistance. So this model can be used to create a real-time representation of what the heart normally experiences. And so we encode this in some hardware. We first scale the force production that's produced by the one-dimensional muscle fiber into its three-dimensional pressure equivalent using a couple of transforming equations. We then pass that immediately on a 20 kilohertz rate through a model of the circulation, and that then predicts the velocity or the distance that the muscle should shorten under those conditions. This is all computed in an FPGA, a field programmable gate array, using LabVIEW Real-Time and LabVIEW FPGA. And now we can get these blue work loops to occur, which look much more like what goes on in the real heart. And this so-called wind kessel loaded work loop produces more work than typical or isometric loaded work loops, which are these flat-topped black work loops. So when we load the heart with a more realistic form of loading, we find that we actually squeeze a little more energy efficiency out of it, because we are now getting closer to the type of load that the heart actually experiences in vivo. And so it is more optimized, one might say, for these sorts of conditions. In this particular sample, it does vary from sample to sample. This sample produced only 10% efficiency using a more traditional dynamometry, if you like, but using our real-time computed wind kessel-based load, we found that the heart was able to produce an efficiency of nearly 15%. So that's, what is that, 33% more efficiency in this particular case when it is loaded more realistically. Now this is very important because diseases of the heart are often associated with, and perhaps arise as the result of, diseases of the circulation. So we can now do interesting things, like, for example, have a normal heart, here shown with its stress length loop in the one-dimensional muscle sample, or the equivalent pressure volume loop of the three-dimensional heart, and now mimic the conditions of hypertension by increasing, for example, the peripheral resistance or the aortic compliance or the circulatory capacitance. And we can mimic conditions of hypertension and hypotension. So this is a really important and powerful tool. You can take normal tissue and expose them to diseased type loads, or you can take diseased tissue and now expose it to a normal load and see how it responds to those changed conditions. So this really is the crux of dynamometry of the heart, to be able to vary the loads while we measure all of the performance, all of the parameters that are of importance that define and describe its pumping ability. And so this is an example here shown of increasing the peripheral resistance, and we're showing how the, these are old data, but we're showing how the stress in the heart's tissues increases gradually as you increase the peripheral resistance. It has to build up more pressure to pump that fluid around the body. So just to sum up, our instrument measures the calcium. I haven't talked about how we measure calcium, but it is using a fluorescence technique, which is fairly standard, and that is done through our optical microscope. The force is measured by a cantilever that's detected by, its bending is detected by an interferometer that can resolve down to 0.3 nanometers. The length of the proteins, or the strain within the tissue, is measured using our image registration technique from stitched together images. Our OCT system allows us to measure the motion of the tissues across many twitches. And sorry, I beg your pardon, that was not OCT, that was the strain field associated with the motion from bright field images, same as the sarcomere length images. And then finally, the OCT information gives us the three-dimensional shape, indeed four-dimensional shape, as it's contracting over time. Now, how on earth do you interpret all of these data? And I'm afraid I don't have time to discuss the complexities of doing so, but it has to involve mathematical modeling. And really at the Auckland Bioengineering Institute, one of the things that we are very good at, in addition to building instruments such as this, is we have some very strong mathematical modeling teams that are highly skilled at taking all of these multivariate data, combining them in a modeling framework where we can take all of the geometric information together with the strain fields, the force production, which can now be scaled into stress, the motion of the various tissues, the calcium that is releasing into the cells and turning on the engine, and model each of these events at multiple scales using mathematical modeling techniques. So that's really all I can say about that process, except to say that it's certainly an area that we're continuing to develop. This is a pretty ugly looking sample, as you can see, but and we're just showing a series of actually body surface meshes. There's no finite element modeling going on here, but certainly efforts are underway to take all of the information and apply them to the mathematical modeling frameworks that we already have in place for describing such soft tissues. So with this information, we can now normalize our measurements of force to known geometrical properties. We can compute material properties for constitutive laws that describe the contraction of the heart and its tissues. We can demonstrate the relationships between these variables and convey all of this information in 4 dimensions. And of course, this one-dimensional, well, it's a 3-dimensional sample, but it contracts primarily along its length, can now be embedded inside a much larger model that describes the entire organ of the heart. And all of those equations can be solved simultaneously. So thank you so much for your attention on this evening in New Zealand, and whatever time it is, wherever you are, whenever you are watching this. Allow me to acknowledge our really talented bunch of postdoctoral fellows, Jinshu Han, Toan Pham, and Kenneth Tran, who each work on this machine, one of our devices that they're working on here. Very talented students, Emily, Jara, and Amy. And our many collaborators, past students who built much of this instrument, particularly Professor Paul Nielsen and Professor Dennis Lazal. Dennis is a physiologist, Paul is a bioengineer and physicist with whom I work And many of these students here, particularly these three, Dr. Alex Anderson, Dr. Callum Johnson, and Dr. Ming Chuk, built the instruments that I've been showing you today, together with help from our engineering staff and other physiologists. Thank you so much for your attention, and I'm happy to take any questions that you might have.
Thank you, Dr. Taberner. Right now, currently, we don't have any questions. If anyone has questions, you can type them into the Q&A, and then Dr. Taberner will answer them live. Currently, no questions. Oh, here's one. Can you see that, Andrew, in the Q&A?
Not yet. No, I cannot.
How different is the saline solution from real blood, is the question from Ray Sperber.
Yeah. Okay, thanks. It's the main difference, I suppose, is its ability to absorb oxygen. Blood, of course, because of all of the hematocrit, can bind oxygen to a much higher partial pressure than the saline solution can. The saline solution contains all of the nutrients that the heart would normally expect, including glucose, of course, to supply the engine, oxygen to keep the cells alive and functioning, and also to take waste products away. But the concentration of the oxygen within the saline is a lot lower than it is in normal blood, perhaps 10% of what you would expect in normal blood. So that limits what you can do with the heart. You can't pace it. Well, sorry, you can pace these tissues at the same rate as they would in the body because they are so small. One of the things about these small muscle samples is that because you've taken them out of the heart, you've also removed them from the circulation that normally keeps them alive, which is actually often running through very small vessels within that tissue. Now the only mechanism for oxygen to get into the muscle is by diffusion through the cells. And it turns out that even with the lower oxygen concentration in the surrounding fluid, enough oxygen can keep diffusing to the center of these cells to stop them dying. So they'll stay alive for up to 12 hours easily while they're actively contracting, and they can indeed be kept in the fridge overnight if you wish to.
Ray has a follow-up question. How does it match with the fluid properties of the blood?
I think in terms of the rheological properties, it's very, very similar. Perhaps probably doesn't have as much viscosity as you might expect in the blood. It of course doesn't clot. So I think in terms of the fluid dynamics, it's very, very similar. And the thermal properties are immeasurably different, at least in my hands. So there's no difference that I can detect from the thermal properties of blood and saline solution.
We have one more question that was put in the chat, Andrew, from Gerard Steinbachen. What is the lifetime of a sample?
Yeah, well, each of these cardiac trabeculae will keep contracting for many, many hours. So a typical experiment will last, it would typically take a student or a researcher up until about 1 in the afternoon. They typically start at, say, let's say 9 in the morning, and they'll do their preparation. They'll get a muscle sample out of the heart at perhaps 1 in the afternoon, mounted in the instrument, and they might do 2 or 3 samples in a day. So the answer to the question is it depends on how hard you are working it. The samples will deteriorate over time if you work them very hard. And so we have to periodically sort of halt the experimental protocol. Let's say we're doing a series of work loops. We might have to halt the protocol and do some something like a fixed length contraction to measure how much force the muscle is producing. And just do that measurement periodically just to check whether the sample is diminishing in force. But typically I would say 4 to 6 hours is a reasonable limit to expect for one of these samples. They can be left in a dish, as I mentioned, and put in the refrigerator overnight, and you can take them out the next day and they will still function perfectly well. One of the really cool things about these samples is you can even put them in a -80-degree freezer and leave them in there for years, take them out, and you can still learn stuff about the engine, despite the fact this tissue has been long dead and frozen. Now, it might seem counterintuitive. When you freeze the tissue, you knock out all of the ability of the tissue to respond to electrical excitation. So you can no longer you can no longer use a spark of electricity to let calcium into the cell. But you can still take that tissue, knock holes in its membrane by using a little bit of detergent, and just put in your own calcium. And as soon as calcium enters in between the proteins of the tissue, it will contract. So you can mimic, in a sense, or you can certainly get the engine to produce force, even though they have been frozen for many years, which I think is really quite neat. It's not quite the same as a living sample, but it's still useful.
Andrew, we have a question on Facebook Live from Chi-Hung Wang. If we excite the muscle instead of applying mechanical force, would the behaviors be different? And then he follows it up with electrical voltage.
Yeah, we do stimulate the muscle with electrical voltage to create a twitch. So perhaps I didn't explain that clearly, and in fact I didn't really tell you about how that happens. So in the slide where I showed you the muscle fiber, it's lying, let's say, along in the horizontal direction. There are platinum electrodes near the ends of the muscle, and we put an electrical voltage which creates a field down the length of the muscle. And that field, it's the voltages that we use are typically less than 10 volts over a distance of about 1 centimeter. That is sufficient to depolarize the muscle membrane and to allow calcium into the cells. So yeah, we can certainly electrically twitch the muscle, and then that's what switches on the force production and gets it to contract.
Okay, it looks like that's all the questions. So thank you so much, Dr. Taberner, for this wonderful talk. And if there's no other questions, I think we'll end the session, and we wish you all a good day, good evening, wherever you are in the world.
Yes, and thank you again for your attendance and your interest in this talk, wherever and whenever you are. And all the best wishes for this very, very demanding and challenging year to all of you, no matter where you are. Thank you.
Thank you.